Good afternoon, or well, good morning, everyone. So this is our pleasure to be here and to share some of our data with you today. So Carole and I, we belong to what we call the Moly Lab or the Molecules team here in Bordeaux, France. And today we're going to tell you a few things about what we have developed in terms of toolbox for the genome engineering of mycoplasma. So in fact, if we go back to history, it's been more than 20 years now that we've been working in developing genetic tools, not only for mycoplasma, but also for other molecules that we're interested in. Especially, we started with spiroplasma, indeed. And we hope someday that we could be able to do that also with phytoplasma. So first we started with developing some RSA replicative plasmids, and then we went to transposon, mutagenesis, and well, as everybody knows here, a main achievement was made at GCVI, especially with John and with Carole, because she was there at that time. And so, well, it's I guess for mycoplasmologists, this was a real change. Yeah, there is a before and after. And but after that, some few things were developed, and especially we developed some new genetic tools to work directly in mycoplasma. And so today we're gonna tell you about a few things about two new tools that we have developed recently, homologous recombination system and the base editor. And then I will leave the floor to Carole, who will update you about genome cloning and genome transplantation. So as everybody knows, CRISPR is a fantastic tool for many organisms, especially eukaryotes, but in bacteria, and many bacteria, it's not that easy to use CRISPR system because most of the bacteria, they don't have a real efficient DNA repair mechanism to deal with the double-strand breaks. And most of the time when you use CRISPR in those bacteria, well, you kill everyone. So, and it seems to be the case for most mycoplasma. So if you don't have this kind of efficient repair system, what can you do? Well, you can try to give it a new one. And this is basically what we have done using the bird pathogen Mycoplasma gallisepticum and using the REC-ET system, which is derived from the SPP1 from Bacillus subtilis phage. And as Carlos presented just before in this day, the first application of REC-T, which is also it's exactly the same as GP35, was developed for mycoplasma pneumonia by our colleagues from CRG. And in our system, we are using a more complete system with not only REC-T, the recombinase, but also REC-E, which is an exonuclease, which is well supposed to improve the efficiency of the tool. And so basically, the idea is to integrate this system in the genome of Mycoplasma gallisepticum, and then to feed the bacteria with a recombination template to trying to get some homologous recombination. So this is an example of a test that we've made on Mycoplasma gallisepticum for a short deletion at that time. And after having integrated the REC-AET expression cassette in the genome of Mycoplasma gallisepticum using a transposon, we used a recombination template, which is represented here with 1 kb homology arms on both sides of the targeted site. And between those arms, you've got an antibiotic marker and also 2 LOX sites. So we make the transformation, we get some transformants. And when we screen them and sequence the the targeted site for some of them, as you can see on the gel. Some of them had the expected profile, PCR profile, and when we sequenced those regions, we confirmed that they had been a precise homologous recombination. So at the moment, this system is functional in Mycoplasma galissympticum, and we have validated it for some short dilation, insertions, and precise replacement. But we failed to dilate larger regions. We tried with the 6.6 kb regions, but we got no homologous recombination. And finally, with the use of the Crelox system, we were able to get rid of the selection marker, which is always interesting if you want to make some iterative manipulations. So now the next goal for us using this tool is to adapt it to other species, other Mycoplasma species. And well, this is currently what we are doing. The second tool I would like to present you is a CRISPR base editor, which is based on deactivated Cas9, which is merged with a cytosine deaminase to get, well, something that will not cut the DNA, but who will drive some precise modification on cytosine in a specific window that it will be well, decided by the guide RNA that is used with the Cas9. So the idea is that this deaminase is able to deaminate some cytosine to uracil, and this could lead us, for example, depending on the design, to a modification of, I don't know, a CAA into a stop codon TAA. So it's quite good to get some gene inactivation. So we first started to use this new tool in Mycoplasma galissympticum, and we integrated using a transposon, a cassette that you can see here, which includes the gene encoding the fusion protein and also the guide RNA to target the gene that we have chosen. So once we have transformed a population of Mycoplasma galissympticum cells, we induced the expression of Cas9 deaminase using an hydrotetracycline. And then we collected the cell population and we evaluated the level of deamination using PCR and Sanger sequencing. And you can see on the right of the screen, it was really efficient, and we were really happy of that because, you know, it was described in the literature for other bacteria, but it was not proven at all for Mycoplasma. So we were quite happy to see something like 100 efficiency. So as you can guess, it was quite easy to isolate specific clones with the mutation that we wanted. So the good news also is that this tool works quite efficiently in other Mycoplasma species distributed all along the phylogenetic trees of Mycoplasma. So we tried it in Mycoplasma mycoides subspecies mycoides, in Mycoplasma bovis, in Mycoplasma agalaxiae, and well, we think it should work in basically any kind of Mycoplasma. So we wanted to finally to test the limit of the system, and for that we choose to target the 55 insertion sequences IS1634 present in the genome of Mycoplasma mycoides subspecies mycoides. And so we used one single guide RNA. And after 3 rounds of induction of the system, we were able to get a conversion of about 70% of the targeted cytosine. And after screening of 20 clones, we finally selected 2 clones with the expected global profile, and we make a whole genome sequencing. And finally, we showed that 54 on the 55 IS were actually inactivated. So with this, it seems now that we've got a toolbox that is quite enriched. And now this is, well, this is the direct in mycoplasma tools. But now I will leave the floor to Carole, who is going to tell you about, well, I guess the ultimate genome engineering tool, which is in yeast.
Okay. So hi everyone. So as you all know, so as part of a synthetic genomic initiative of generating a synthetic cell, the GCVI team reported back in 2009 a set of new technologies for manipulating molecules' genomes. And those technologies included in-yeast cloning of whole bacterial genome in the form of yeast centromeric plasmid, followed by in-yeast genome engineering using the large repertoire of yeast genetic tools, and finally the transplantation of the modified genome from yeast to a recipient cell to get a cell controlled by the incoming genomes. So in fact, during the remaining few minutes, what I want to do today is just to give you an update about these three major steps. So I will be a little bit quick. So first step is in-yeast cloning of whole bacterial genome. So following the work that was done in 2009 at GCVI, so during which, in fact, the elements that were required to maintain the whole genome in yeast were inserted in the bacteria of interest by transformation. Many other methods were developed and described in the literature. So they allow to clone either they allow to clone either native genome from genetically tractable bacteria or native genome from intractable bacteria or synthetic genomes. So recently our team has developed two new methods. They are presented at the top. They allow the cloning of native genome from any kind of bacteria. And the advantage of those methods is that they allow, in fact, to clone and engineer in one step a 1 megabase genome into yeast, because the yeast, in fact, are preloaded with the Cas9, CRISPR-Cas9 system. So using those tools, to date, 21 native genomes from 15 different species of molecules are cloned into yeast. So they are well distributed all along the molecules phylogenetic tree and among the four phylogenetic group of mycoplasma, as you can see here. So they are noted in bold. Their size range from 0.5 to 1.8 megabase, and the GC% is from 24 to 40%. Okay, so the second step is the in-yeast genome engineering. So here, as maybe you know, yeast, they are endowed with efficient and well-established method for DNA manipulation. However, genetic engineering method classically used in yeast were revisited before being applied to bacterial genome. But this allowed the development of new methods, new techniques. I won't describe them all, so just give the name is the tracking method, and more recently a system based on the CRISPR-Cas9 editing tools. And all these tools allow to perform all kinds of modification in yeast, which means from gene deletion to gene insertion, replacement, single point mutation, meaning that we have a wide range of tools which offer unprecedented perspective towards the engineering of bacterial genome cloning yeast. So last step. the last important step of this cycle concerns the transplantation of the bacterial genome from yeast into a recipient bacterium. So if you remember, back in 2007, in fact, the first transplant were obtained at GCVI using the mycoplasma capricolum as a recipient cell. So it's the one in purple here, and using the mycoplasma mycoides subspecies capri as a donor genome. From there, in fact, now we have 11 new genomes originating from 7 different species that are currently transplanted in MCAP. Most of them are ruminant mycoplasma of the so-called mycoides group, but we have also other species, more distant molecules like Mycoplasma florum, and this work was done with a group of Sébastien Rodry in Canada. So this work allowed us to develop new engineering platform for very important pathogen. This is one point, but also this work allowed us to provide us with new information, very important for the whole genome transplantation process. First of all, we noticed that the phylogenetic distance was very important. It is a key factor. So the highest transplantation efficiency were obtained with species, with the genome of species that are very close to MCAP. And then the efficiency is diminished with the increase of phylogenetic distance. We also reached a limit of compatibility between 83 to 90% of similarity of core proteome. And finally, we also noticed that we had to face to a species-specific factor, and we had to deal with that. So the next challenge for our team, we want to go further. We would like first to understand the molecular mechanism underlying whole genome transplantation, and in particular to better understand the rules of compatibility and push back the current limits of genome transplantation. And second, we would like to implement this genomic engineering method to other bacterial species, molecules and non-molecules, as John. I think here we have a common objective. So this second question, actually, before to dive into this point, we had a question. We wanted to answer one question: is MCAP the only possible recipient cell? And in fact, our current work showed that the answer is no, and at least 2 other species can be used as a recipient for whole genome transplantation. So the success of whole genome transplantation still relies on the PG protocols, but what we noticed is we had to work very very strongly on the protocols. They are all different. For instance, the protocol that work with MCAP as a recipient cell is different from the one from Mycoplasma leachii, or or the one of Mycoplasma mycoides subspecies mycoides, meaning that we have 3 recipient cells for now, but 3 very different protocols. So on one hand, this is okay, we work here with species that are close to MCAP, so not very far away. But on the other hand, it was very comforting for us to obtain this kind of results, because it means that other cells than MCAP can be used as a recipient for genome transplantation. So now that we know that, we in fact collected a good amount of data, and we have undertaken the development of whole genome transplantation for new pair of species. They are noted or presented in blue or red in this phylogenetic tree. Some of them, in fact, are from different phylogenetic groups, as you can see, and some are non-molecule species, as Bacillus subtilis. Just to finish, in fact, about the whole talk, we made a summary with Pascal. So this table, if I can say, represent all the tools that are available for molecule species to date, both the in-yeast genetic tools and in mycoplasma genetic tools. And hopefully, these tools can be applied to the minimal SIN 3.A or 3.B. And here, just we want to thank the Molecule Lab, particularly the synthetic biology group, and also our partnerships and fundings, among them John, and also Sébastien, Luis, Maria. And I want to thank Bougamit Karas also, because we started very interesting discussion. And here are some of our mycoplasma friends. Thank you.
Carole, Pascal, I'm dazzled again, much like I was when I heard Maria's group talk about the new tools that they've made. So the mycoides group really doesn't do recombination very well. Does that affect its ability to use to work? I have to go back to your slide a couple of slides ago, but does that affect the things you can use with that organism?
Well, in fact, I have to remind some work that Carole did when she was in PhD some years ago. And well, she noticed that, when you used Orycea plasmids, one question that was discussed just before is, will this plasmid stay as an exogenous replicative form, or will it integrate? And we noticed that depending within the mycoides cluster, there were some differences. Maybe, Carole, you can precise this.
Yes, notifiable differences because, for instance, Orycea plasmid in MMC has a tendency to integrate the genome very quickly after just 1-2 passages in vitro. In contrast, species like Mycoplasma capricolum keep the plasmid free in the cells during, I think I never saw the plasmid integrated into the genome for MCAP, for instance. So it means there are differences.
But it's non-homologous recombination.
Yeah, it's difficult to say.
Well, what it happens with MMC, the integration is at the replication origin, so it's kind of homologous recombination.
But I'm thinking about the mycoides strain that we produced here at the JCVI, where there were two Oryces in it, and it was a very unhappy cell, as I remember.
This is true. Yeah, we had weird results with this one.
And then Gallisepticum has evidence of its own CRISPR system.
Yeah, it has its own. Yeah.
Have you thought about trying to use that as a tool in mycoplasmas?
Well, in fact, we have used it, but as a counter selection marker, because the problem is that if you feed the Mycoplasma gallisticum with a self-targeting guide RNA, you kill everyone, and the only cells that you recover are mutants of the system. So it seems that there is no mechanism for double-strand break repair that are efficient enough in Mycoplasma galliscepticum to avoid lethality. So, I think it is possible to use the system, but you can use it as a scissor. Nothing much than that, I guess.
So in Gallisepticum, is it designed to kill the cell, or was it designed to get rid of phage and not kill the cell?
Oh, I guess it's, as every natural system, it was well designed by nature to help the cell fighting against invading phages or mobile DNAs and some things like that, obviously. And we, when we have a look on the CRISPR arrays in the genomes of Gallisepticum, it's sometimes we find some spacer that are that are looks like phages, so it makes sense.
Yo, you look like you have a question.
Well, how did you find the three?
Introduce yourself, please, Yo.
Yeah, I'm Yo Suzuki. I work with John. I'm an assistant professor at JCVI. How did you find the three recipients cell types, and then how would you? What features do you look for to find the fourth recipient?
And with that question, what is different about the protocols, if you can say?
Yeah.
Many things. We had
Does it still use calcium?
It still uses calcium, but in fact, we had to work out on many parameters. And so including the wash buffers, so it uses calcium, also the pH percent, the time of contact between the PG and the cells. We also something that is very important, and I think it's so I don't know if it's a technical or biological hurdles, but frankly, for a while, for one species, it was impossible to get results. But in fact, we work with the wrong antibiotic marker or the wrong promoters and the wrong plate, the medium. And and once you have all this, in fact, it worked. But you have to find out which medium to use to grow the cells to plate, which antibiotic you want to use, which concentration of antibiotic to select your transplants. And okay, there are many parameters, so it's a very long work. But once you have all this, in fact, you just realize that you can make it and it's possible.
Sebastian.
Hi, Pascal and Carole. Great to see you. I was just curious, Carole, about the different recipients that you mentioned. You said that the protocol is different, but I was curious about the efficiency. Do you recapitulate the efficiency that you can get with Capricolum when you use other recipients, or is it better or is it worse?
So I would say, so it's maybe difficult to compare, but for one species, Mycoplasma lichii, we put a little bit less effort on that one, but we could recover transplant each time. For the other one, Mycoplasma mycoides, subspecies mycoides, we worked very hard on the protocol, and we have actually excellent efficiencies right now. Even I would say even better than in MCAP. So yes, maybe it depends on the effort that you put on the and the time, because it requires a lot of time.
Thank you. And just you already know this, but we identified one Mesoplasma strain that is really good at getting OREC plasmid and transposon, so 100-fold better efficiency. So would be nice to try these eventually. Yes,
with pleasure. And saying this, I think another very important point is the choice of the strain. You have to try different strains. And I think we make all an error here talking about species. We should talk about strains. I think strains behave differently regarding transformation, transplantation, cloning. Even for yeast cloning, the strain that you use is very important.
And susceptibility to antibiotics
and
also is very
sensitivity
different.
to antibiotics. And so the question of the strains that you want to use is very important. Just be careful about that.
Other questions?
Beyond,
go ahead.
Extending beyond mycoplasmas is a major challenge.
To go behind mycoplasmas?
Beyond,
yes,
yeah.
Yeah, John will talk about that. We started also a project with Bacillus subtilis, and actually it is a huge challenge, mainly because, it's a gram-positive bacteria with a large genome of 4 megabases. So we're working with a strain with 3 megabases genome. It's a little bit less large. And so right now, in fact, we succeeded in cloning the whole genome in pieces, but not entirely. And we trying to do more effort to get it complete into yeast. And in parallel, yes, we are working on the transplantation protocol also for Bacillus subtilis. But here, for Bacillus subtilis, I think the first barrier to overcome is the recombination barrier, because as John mentioned, with mycoplasma, the advantage to work with mycoplasma is they do not recombine so frequently. And gram-positive bacteria as Bacillus subtilis recombine a lot. The frequency of homologous recombination is really high. So when you want to transplant a new genome into the recipient cells, in fact, they have a tendency to recombine a lot. So we have to overcome this barrier.